Hello, everyone, and welcome to today's live broadcast. Differentiation sensitizes stem-like glioblastoma tumor-initiating cells to mechanical inputs, presented by Jasmine Hughes, a PhD candidate in the UC Berkeley UCSF Graduate Program in Bioengineering. I am Susie Valdez of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Thermo Fisher Scientific. For more information on our sponsor, please visit them at thermofisher.com. Now let's get started. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Answer a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I'd now like to introduce our presenter, Jasmine Hughes. Jasmine Hughes is a PhD candidate in the UC Berkeley UCSF Graduate Program in Bioengineering and is working in the laboratory of Professor Sanjay Kumar at the University of California, Berkeley. Jasmine is interested in how cells sense and respond to the mechanical properties of their microenvironment. She answers open questions in mechanobiology by designing and applying novel synthetic biology and systems biology tools. She's particularly interested in how mechanically regulated signaling is perturbed in glioblastoma stem-like tumor-initiating cells. Jasmine received her bachelor's degree in chemical engineering from McGill University and has been recognized for her work by the Natural Scientists and Engineering Research Council of Canada and by the Siebel Scholars Foundation. In addition to her academic pursuits, Jasmine also played a leadership role in advancing professional development opportunities for Berkeley PhDs through the nonprofit organization Beyond Academia. Jasmine, you may now begin your presentation.
Thank you for the introduction, and thank you to all of you guys for tuning in. I'm really excited to talk about one of the projects that I've been working on for my PhD, where we're particularly looking at glioblastoma tumor-initiating cells. So I'll get into that in a little bit more detail. I am actually a chemical engineer by training, and I think that that background has given me a different way of looking at cancer cells from sort of like a force balance perspective. And I really enjoyed the biology parts of my chemical engineering training, so I switched into bioengineering. So I'm excited to talk to you about, I guess, how cells sense forces and things like that. So glioblastoma is the most common and lethal primary brain cancer. It's an aggressive malignant cancer of glial cells. In the last 15 years, we've made a lot of technological advances. So this includes the introduction of temozolomide and more aggressive tissue removal surgeries that are guided by improved imaging technologies. However, recurrence is near universal despite these technological advances and radiation therapies, leaving median survival time at just about 15 months. So one of the problems that we have when we're treating glioblastoma is that the cells are able to migrate through the tissue, which makes it very difficult for us to surgically remove. These cells are able to then reestablish the tumor, which is what you see sort of at the borders of the tumor resection, sort of in the 12 months, 17 months images here. And this process is ultimately fatal. So part of what's thought to govern this tumor reestablishment is the presence of tumor-initiating cells, which are sometimes also called tumor stem cells. So these cells evade treatment, invade through the tissue, and recapitulate the original tumor. So these cells have been shown to migrate along white matter tracts in the brain and along blood vessels and other structures in the brain that have differences in mechanical properties. And that's led to some people to wonder how these cells sense the mechanical properties of their environment. So we've looked at a lot of, we've identified key genes and soluble factors, and this has led to a lot of insight into how to treat these cells, but there's some thought that maybe if we better understood the mechanical sensitivity process, we might better understand how to treat these cells. So one of the key techniques that we use in this field to look at how cells sense mechanical cues are the use of things like polyacrylamide gels and other biomaterials, where we can vary the mechanical properties of them by varying their composition, how they're cross-linked. So one of the seminal works in this field came from now over a decade ago, when Paczek and colleagues discovered that matrix stiffness perturbed tissue structures, such as in these acini shown here. These are MCF10A cells in a 3D gel culture. And this increased matrix stiffness promoted a more malignant phenotype. So this work and the work by many others in the field have led to this model shown on the right, in which genetic mutations in tissue mechanical structure produce a positive feedback loop that leads to malignant tumors. So this process can begin with an oncogenic mutation, so that's the left of this diagram, and this mutation leads to how changes in how cells sense mechanical cues or how they remodel the extracellular matrix in their environment. Or alternatively, on the right side of this diagram, this process can begin through changes to the normal structure of the extracellular matrix, such as from chronic fibrosis, leading to the acquisition of oncogenic mutations. So regardless of which path you get to begin with, you then get this sort of feedback loop of mechanical signals and genetic mutation. And this creates an environment that promotes the growth, survival, and invasion. So over the last couple of decades, we've greatly increased our understanding of how cells sense mechanical changes in our environment. And these are largely thought to occur through actin-myosin contractility. So cells have an actin-myosin cytoskeleton, so filaments of actin cross-linked with myosin 2. And cells are constantly pulling on their environment. And this force that's generated by myosin motor proteins is transmitted to the extracellular matrix through complexes or contact points known as focal adhesions. So this force balance between the extracellular matrix and the force generations in the cell is interpreted by the cell into a diverse array of phenotypes that includes cell shape, proliferation, and migration. So one of the major signaling pathways through which the cell contractile forces are regulated is the Rho pathway, where Rho A activates myosin light chain through phosphorylation, and this leads to myosin being able to exert a contractile force, and that leads to mechanosensing. So typically, soft substrates, such as the brain, are translated by the cell into reduced migration and reduced proliferation, which acts as a bit of a stop signal. So it's kind of an interesting question for in glioma, where the brain is obviously, or glioblastoma, where the brain is obviously a very soft substrate. So how is that influencing glioma cells? So a few years ago, we took a glioblastoma cell line, U87s and 273s, and we put them on gels of different stiffness. So very soft, which is 80 pascals on the left, or very stiff, which is 119 kPa. As stiffness is higher than this, we actually wouldn't see much of a difference in phenotype. So what we found is that in agreement with findings in most other cell types, these cells showed a very strong sensitivity to mechanical signals. So on the very stiff substrates, they had these very spread morphologies. They had very prominent actin-myosin stress fibers shown in green, and they had prominent focal adhesions. Those are the contact points to the extracellular matrix. On very soft substrates, they had that rounded morphology. They didn't have prominent stress fibers. They had very small focal adhesions. So this was also true for other behaviors that are known to be mechanically regulated. So cell speed, for example, is very strongly mechanosensitive, and proliferation, so as measured by incorporation of BRDU, was also very mechanosensitive. So these were continuous cell lines, and that's important when you're thinking about cancer biology, of course, because these cells are cultured in serum, and they've been passaged many times, and are at this point fairly far removed from physiological brain tumor cells. In fact, if you put these cells into a mouse brain in a xenograft model, they don't really even form classic glioblastoma-like tumors. They're kind of more circumscribed, compact, smaller tumors. So because of these differences, we wanted to make sure that this also still upheld at a more sort of physiological model level. So we turned to patient-derived stem cells. So this was a collaboration with Brent Reynolds' lab in Florida, and they're cells that have been harvested from donated, surgically removed glioblastoma tissue. So we select these cells by growing them in a neurosphere suspension culture media, and this is supplemented with a high concentration of epidermal growth factor and fibroblast growth factors, so EGF and FGF. So these cells express stem markers, like SOX2 and nestin, for example. But particularly importantly, if we were to xenograft them into mice, then these cells form tumors with the classic hallmarks of glioblastoma. So that includes areas of necrosis, diffuse migration through the brain tissue around it, to palisading structures. So when we put these cells on our gels, the very soft ones and the very stiff ones, we noticed that they actually didn't form stress fibers on either stiffness particularly. They didn't have differences in their focal adhesions. And more importantly, they're kind of similarly spread on all of the different stiffnesses. We then looked at migration as well. And it was still a little bit mechanosensitive, but not to the same dramatic effect that we'd seen before. So they were still fairly able to migrate on soft substrates. We also looked at proliferation, again with incorporation of BRDU, and we also didn't see a mechanically regulated difference in proliferation. So because these cells weren't forming the stress fibers and the focal adhesions that we normally expect to see from cells generating contractile forces on stiff matrices, we wondered if perhaps these cells were generating smaller amounts of contractile force. We wondered if we were able to change that. So we turned to the ROC pathway, and we thought, what happens if we increase the amount of myosin light chain phosphorylation? So we took a doxycycline inducible cassette, and we genetically engineered our cells to be able to induce expression of RhoA. This is actually a constitutively active mutant of RhoA, so it's always on in the cells. And we found that when we activated RhoA by introducing doxycycline into the media, we were able to increase the phosphorylation levels of the myosin light chains. And when we put our cells with the activated RhoA machinery, we found that we could induce a level of mechanosensitivity, which was really interesting to see. So these cells, the control cells, were equally spread on the soft matrices and on the stiff matrices. But the constitutively active RhoA cells with the activated myosin contractility were much more round on the soft gels compared to on the stiff gels. So this also held true for migration as well. And we then wondered if that would lead to differences in invasion as well. So we did a three-dimensional migration assay. So looking at how the cells move through three-dimensional collagen. So the control cells, when you put a little spheroid into three-dimensional collagen, they start to invade pretty well. They're fairly invasive cells. And in contrast, the constitutively active RhoA cells, they remained very tight into their sort of neurospheres. They didn't invade through the matrix. We next looked at transwell migration, which is also sort of a metric of how well cells are able to invade. We found that by activating the RhoA pathway, we could reduce the amount of migration through a transwell. So this was kind of promising for, cancer biology reasons. And we wondered if this would also hold true at still a more physiologically relevant model. So we turned to the xenograft that we've been doing before. So the control cells formed these classical glioblastoma tumors, large with diffuse migration away from the core, and also some migration to the other side of the brain. In contrast, the RhoA cells formed much smaller and circumscribed tumors with very clear boundaries, and we didn't see establishment of secondary tumors. This translated to a much smaller amount of tumor occupancy in the brain and extended mouse survival time. So what we have here is sort of this observation that these cells are not mechanosensitive, and that by increasing the activity of the actin-myosin contractility, we could increase the mechanosensitivity and also decrease tumor migration. So this made us wonder, because of this really striking relationship between mechanosensitivity and invasion, we wanted to better understand the relationship between stemness and mechanosensitivity. So is this mechanosensitivity property linked to the fact that these cells are sort of stem-like? So one morphogen of particular interest is bone morphogenic protein 4, which has been established as a signaling cue that's important in neural development. And it also plays a role in the adult neural stem cell niche. So this is a region that is thought to give rise to a sizable proportion of glioblastomas. This is of particular interest for glioblastoma tumor-initiating cells. So another lab has looked at the role that BMP4 plays in GBM tumor-initiating cells. They found that if you treat the cells with BMP4, they stop forming neurospheres, which is an indication that they lose some of their stem-like properties. But also importantly, they show an increase in the expression of differentiation markers. So for example, they have shown an increase in GFAP, which is an astrocyte marker, and an increase in beta-tubulin 3, which is a neural marker. I also saw an increase in GAL-C, which is a oligodendrocyte marker, although that's to a smaller extent. Also very interesting, when you treat these cells with BMP4, they stop being able to form these glioblastoma tumors. So the tumors that do form are very small and compact. So we wondered what happens if you treat our cells with BMP4. Do they retain their mechanoinsensitivity, or do they now become able to contract their environment and interpret those signals? So what we found is that when we treat our cells with BMP4, they actually do become a little bit more mechanosensitive. So in general, the cells are smaller when treated with BMP4, but they become able to sense the differences in stiffnesses. So now they're rounded on soft matrices and spread on stiff. That's sort of the classic phenotype that you'd expect from mechanosensitive cells. We wondered if this would also translate to other readouts of mechanosensitivity. So one that's become increasingly used in the field is translocation of mechanosensitive transcription factors. So this is sort of a hallmark paper in the field where they found that the transcription factors YAP and TAZ go to the nucleus on stiff substrates, but on soft substrates, like the one on the right here, they tend to be more cytoplasmic. So this has been replicated by a number of labs, although I think for most people, the difference isn't quite as clear-cut in most cell lines. So it's true for ours as well. We've looked at neural stem cells. They behave very similarly. So what we found is that for the growth factor-treated cells on the soft or the stiff matrices, their TAZ localization was mostly cytoplasmic with a little bit of nuclear staining as well. However, on the stiff substrates, we've had an increase in nuclear localization. So particularly, you can see that on the bottom right of these cells here. So this suggested to us that there was perhaps some role that transcription was playing in our cells on the stiff matrices. So we wanted to know kind of what was going on in these cells. What's changing when they differentiate? Is there a transcriptionally regulated reason these cells might be more mechanosensitive? So to get a sort of a snapshot idea of what these cells were going through, we did RNA sequencing. So we looked at cells on either the soft matrices or on the stiff matrices, and that had been treated either with the growth factors or with BMP4. So we did a 1-week differentiation. So they should be fairly differentiated at this point. So what we found is that BMP4 elicits pronounced transcriptomic effects. So I don't think that's particularly surprising since we are sort of sending these cells into a differentiated condition. So this is a volcano plot. So it's log fold change on the bottom and then adjusted p-value on the y-axis. And the orange dots are the genes that have been differentially expressed. So what we found is that there's a very large number of genes that are similarly influenced by BMP4 regardless of stiffness, which you can see in the Venn diagram on the left. There's also some genes that are unique to cells differentiated on soft, and some genes are unique to those differentiated on stiff. Particularly interesting to us is the effect of mechanical properties of the environment. So these volcano plots show the difference between the stiff versus soft for BMP treated on the left, and then on the right is for the growth factor treated cells. So what we found is that there's sort of a threefold increase in the number of transcripts that are differentially expressed on soft versus stiff for the BMP4 cells compared to the growth factor treated cells. So that tells us that yes, in fact, BMP4 sensitizes these cells to mechanical input. I think what was kind of surprising to us, however, is that there's very few genes that were mechanosensitive in both the growth factor treated condition as well as the BMP4 treated condition. Most of these were higher expressed on stiff, so these are the genes shown in orange. We also found that we had the sort of expected changes in stem markers like OLIG2, and we found the expected changes in differentiation markers like beta-3 tubulin and GSAP. There were a couple markers that we looked at that we expected to see changes in, so that's particularly SOX2 and GAL-C. So SOX2 is a stem marker. There's been some literature that SOX2 expression doesn't actually change during differentiation of glioblastoma tumor-initiating cells. And it's thought that even despite pro-differentiation signals, these cells still try to retain some of their sort of stem cell plasticity, and that's important for cancer progression. So while this finding was initially surprising to us, I think that it's backed up by other groups as well. And then we didn't see changes in GAL-C, which is an oligodendrocyte marker. So from prior papers, we've also noticed that this doesn't change that much during differentiation of these cells, so it wasn't too surprising that we didn't see significant differences in RNA-seq either. So we wanted to get an idea of which pathways are being changed by BMP4 and if that could explain any of these changes in mechanosensitivity. So what we found is that we looked at the pathways that are overrepresented in the genes that are significantly differentially expressed after BMP4 treatment. We found that ECM receptor interactions came up at the top. So this is kind of interesting to suggest that BMP4 influences how the cell is tripping onto the environment, how it's producing environment around it, for example. We also found that the genes that are differentially expressed particularly tend to localize to the stress fibers, focal adhesions, and cell adherence junctions. So all of this really supports a model where this BMP4 differentiation influences how cells are gripping onto their environment, and that could maybe explain some of the more phenotypic differences that I've shown previously. I think sort of as mechanobiologists, we were particularly interested in what changes as a result of mechanosensitivity. So we did the same sort of analysis looking at what happens or how the genes that are differentially expressed by stiffness, like what pathways we see there. So we looked and we found that ribosomes and genes related to oxidative phosphorylation are particularly enriched in this gene set, as well as a couple other diseases. So Parkinson's disease, Alzheimer's disease, and Huntington's disease are all diseases of oxidative phosphorylation. So I'll kind of group those into my discussions going forward. The genes that are overrepresented in mechanosensitivity also tend to localize to ribosomes, to the extracellular matrix, to focal adhesions, and to the mitochondria. So again, that's kind of reinforcing sort of this idea that differentiation influences how cells are gripping on their environment, and that mechanosensitive or mechanical cues influence, I guess, energy production and translation. So what we noticed is that oxidative phosphorylation genes tend to be downregulated by BMP4 and upregulated on stress substrates, while ribosomal proteins are upregulated by BMP4 and on stiff substrates. So we wanted to verify this at the protein level, so we did some Western blots. So we picked a couple of candidate genes that were particularly associated with the pathways that we'd seen before, so cell ECM communication. So we looked at alpha-actinin-1 mapped to kinase, TAZ, and Zixin, and we saw the gene expression change in the direction that we had expected. So it was encouraging. We also looked at integrin alpha-2. I think this is particularly an interesting marker because it's upregulated by BMP4, and its transcription is also different at different stiffnesses. So we looked to see if that held true also at the protein level, and we largely saw that. And we looked to see how that localized around the cell. So we did see an increase in expression in integrin alpha-2 in BMP4 treated cells, and also on the CIS substrates, it tended to localize to the cell exterior, to cell-cell contact, which is consistent with sort of mechanosensitivity, with general mechanosensitivity. So normally you see focal adhesions clustering where the cell is sort of exerting tension on its matrix. So this is suggesting that the integrin alpha-2 is in fact at cell borders. I think we're particularly interested in changes in integrin signaling because integrin signals, changes in integrin expression is also strongly associated with cancer progression. So one of the questions that we had is what transcription factors are governing these differences in mechanosensing? So what the field has generally coalesced around is the idea that YAP and TAZ are these key transcription factors that govern mechanosensitivity. So what we were interested in is that YAP-TAZ target genes actually don't seem to be changing all that much on different stiffnesses in the differentiated cells. So we did see a few genes that are YAP-TAZ targets change in the growth factor, sort of stem cell-like cells. So we kind of wondered what transcription factors could be governing these changes if it's not YAP and TAZ. So we looked to see what sequences were upstream of genes that were mechanically regulated and what transcription factors bound to those sequences and which of those transcription factors were overrepresented. So we found this short list of genes. We then wanted to know, do any of these have relevance for cancer? Because we are, of course, studying glioblastoma. So we turned to TCGA, which is an online resource that has a lot of clinical information that's very well curated. So we like to see which of these transcription factors seem to be of interest for glioblastoma. So we particularly honed in on these two, TSCP2 in particular, because this was mutated in a fairly high percentage of cases, and also it was often downregulated. So this is, of course, of interest because if there was a transcription factor that is important for mechanosensing, we might expect that if it's downregulated, the cancer might be more aggressive because we have this sort of model where this loss of mechanosensitivity results in an increase in invasion. So TSCP2 is downstream of a number of oncogenes. It's got a number of names, so everyone seems to know it under a different name. And it also plays a role in chemoresistance and EMT and a number of phenotypes. So its expression is also, or mutations in this gene also influence survival. And there's a correlation between its expression levels and how long the patient survives after the initial diagnosis. So I think we're pretty excited to further probe this. We have some experiments in the works. So I'd like to refresh what we've been discussing. So tumor-initiating cells, so these are stem-like cells that are isolated from glioblastoma tumors. They are surprisingly insensitive to mechanical cues. However, we're able to genetically engineer these cells to activate the cell's contractile machinery, and this increases the cell's mechanosensitivity, as well as reducing their ability to invade through two-dimensional structures and through small pores. So we can sensitize the cells to mechanical cues, and this inhibits the tumor progression in a mouse model. However, this isn't the only way that we can alter how these cells process mechanical cues. We can also look at differentiation. So differentiation also changes how these cells are able to interpret mechanical cues. BMP4 upregulates alters cell ECM communication proteins. And BMP4 treated cells show changes in ribosome protein and oxidative phosphorylation genes. Interestingly, the transcription factors that seem to be governing these changes aren't canonical mechanotransduction transcription factors, so YAP and TAZ, and we've been looking at a couple other transcription factors that do seem to be mechanosensitive. So with that, I'd like to thank my lab mates, particularly my PI, Sanjay Kumar, and the undergraduate students that are working with me, on, particularly Jeanette, on this project, and of course my lab mates. And with that, I'd like to open it up to any questions that we have about what I've been talking about.
Thank you, Jasmine, for that informative presentation. We will now start the live Q&A portion of our webinar. If you have any questions you'd like to ask, please do so now. Just click on that Answer a Question box located on the far left of your screen. We'll answer as many questions as we have time for. So let's get started and take a look at our first incoming question. Our first question, Jasmine, is, you primarily looked at changes in TAZ localization. Do you also see differences in YAP nuclear translocation in your cells on different stiffnesses?
Hi, yeah, so that's a really interesting question. We've done YAP stains in our cells. We don't see as strong a difference as with TAZ, and this is actually also true for a couple different cell lines that we've tried it on. So neural stem cells is another cell line that our lab uses fairly thoroughly. And we find the TAZ difference tends to be stronger than the YAP difference. And I think in talking with a few other people in the field, that seems to pan out in several other cell types as well.
Thank you for that. Our next question coming in from our audience is, have you looked at the effect that other myosin light change activators have in regulating mechanotransduction in these cells?
Yeah, so I used the row A because we used that most thoroughly. That's what we did our mouse studies with, but we've also done the in vitro work with a couple other row A pathway proteins, so ROC, and then also MLCK, which works orthogonally to row A. So that, and in combination with blebbistatin and similar myosin light chain inhibitors, let's both say fairly confidently that the phenotypes we've seen when we activate row A happen through actin-myosin contractility and not through other pathways that row A can act on.
Thank you for that, and thank you for our audience members and for their participation. Our next question coming in from our audience is, are all glioblastoma tumor-initiating cells mechanosensitive?
That's also a really good question. So we did our work in two cell lines, both from classic GBM subtypes, and we found that both of these cell lines were mechanosensitive. However, other labs have done larger panels of tumor-initiating cells, and there seems to be some heterogeneity. So some tumor-initiating cells show completely normal sensitivity to mechanical cues. However, others do show this mechanical insensitivity that we've seen, and particularly interestingly, the cells that show the least mechanosensitivity tend to also be the most invasive. So it doesn't seem to be a universal property, but it does seem to be correlated with how able these tumors are to spread through tissue and to propagate new tumors.
Thank you. And have you looked at the effects of other differentiation cues in addition to BMP4?
So we've done some studies with serum just to show that this has a similar effect in terms of, increasing in differentiation markers and reducing some markers. But serum differentiation tends to be a little bit more heterogeneous. You get some batch-to-batch variability because it's not always really clear what components are in each batch of serum. So we stuck with BMP4 because it does have physiological relevance to the niche that these cells live in, in bodies, and also because it's, one cue, so it's a little bit perhaps easier to figure out what the effects are.
Thank you for that, Jasmine. And do you have any closing remarks that you'd like to provide for our audience today?
No, I guess if you have further comments or you want to follow up with me, I'm available on Twitter. So I put my handle in the beginning, that's at Jas Hughes, JAS_HUGHES. I'm happy to hear from people later, or you can find my email on our lab website as well.
Thank you, Jasmine. And I want to remind our audience members that if they have any additional questions, they can email you. Before I go, I want to thank them for joining us today and for their interesting questions. We also want to thank you as the presenter, and we want to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. This webcast can be viewed on demand through September 2018. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, everyone, thanks for joining us. Goodbye.